threshold yet. Just before we reach threshold, at this particular current, voltage-gated calcium channels will open. These are called transient channels, or T calcium channels, because they only open for a brief period, right? So that would be this part right here. Voltage gated. But that allows the cell, this is calcium coming into the cell, and again it opens because of the voltage due to the rising amount of sodium inside the cell, and that brings the cell to threshold. Okay? So they open and close very quickly, hence the term transient. And then we actually reach threshold. And then this particular component here, now we're at our action potential. So this occurs due to long-lasting calcium channels. They open more quickly, so they're also known as fast calcium channels, and they stay open longer, hence the long-lasting. So unlike our cardiac muscle, and unlike our skeletal muscle, this depolarization of the action potential is due to calcium, not to sodium. So calcium becomes more important in the heart than it is in skeletal muscle tissue. So as calcium comes into the cell from extracellular fluid, it binds to potassium channels, and that causes the potassium channels to open. Okay? Voltage-gated potassium channels will open. And what is that going to result in? Repolarization. So these are voltage-gated channels. And then let's see if I can find another color. So the calcium binds to the potassium channels, and voltage-gated potassium channels will open. And we see repolarization. This rush of positive ions into the cell doesn't just stay in our autorhythmic cells, but it leads through gap junctions to adjacent cells, our conductive cells, as well as muscle cells, and we see a wave of muscle contraction. That's how the muscle cells are triggered, right? Are through these positive ions leaving via gap junctions into the contractile cells. But we'll come back to that when we talk about contractile cells. So the potassium channels open, potassium diffuses out. As repolarization occurs, that causes the calcium channels to close, which further increases repolarization because now positive ions are no longer coming into the cell. And we continue repolarization. So we're losing positive ions and we're no longer gaining the calcium ions. Similar to our muscle contraction, except it was sodium and potassium. And now it's calcium and potassium. longer gain the positive calcium ions, and we're losing the positive potassium ions, so we see repolarization. And as the cell continues to repolarize, now it's hyperpolarizing, all right? And that's going to trigger the opening of those funny channels, and we start depolarization again. Okay? So we never actually see a flat resting membrane because the hyperpolarization triggers the depolarization again. So those funny current channels are voltage gated. They can open and close. Sodium leak channels are always there, right? But we modulate the rate of depolarization and how fast these autorhythmic cells will depolarize with those funny current channels. Leak channels are always there. They don't have gates, so we can't change that, but we can change those funny current channels.
Can you say it again? The leak channels,
So the leak channels are always present. They don't have gates, so we can't open. We can add more leak channels, but that takes time. So when we try to up here, when we try to change the rate, so here I'm talking about increasing the rate. So norepinephrine will bind. I'll diagram this on the board. It'll make it a little bit clearer. Norepinephrine, which is our sympathetic neurotransmitter, will bind to receptors. It's a G protein pathway. And so what it does is G protein activates the enzyme adenylate cyclase. Adenylate cyclase converts ATP to cyclic AMP. Cyclic AMP binds to those funny current channels and makes them open earlier. So the cell doesn't have to hyperpolarize as much. I'll diagram this on the board so it's not just words, okay? It also causes those long-lasting calcium channels to open earlier and to stay open longer. So what we see is we see this action potential occur more readily. So let's look at that. Now, I think that will answer your question with pictures rather than words. So, sympathetic effect. Norepinephrine is released, okay, and binds to beta-1 receptors on the SA node, say. So here it is binding to beta-1 receptor, and that's going to activate a alpha subunit of the G protein, which binds to adenylate cyclase, forms cyclic AMP, which opens the cyclic AMP itself, can bind to the funny channel, and that will open earlier. So here in our normal diagram, funny channel here, the funny current channel would open at a minus 50 to a minus 55. In other words, we had to come down all the way to here before the funny current channel would open. With norepinephrine and cyclic AMP binding to the funny channel, it will open at this point. The cell doesn't have to lose as much of the potassium before the funny current channels will open. Remember, the funny current channels are voltage gated. they're opened by negative force rather than a positive. So typically when we talk about voltage-gated channels, enough positive ions have to enter the cell before the channel changes shape. In the case of the funny current channels, enough positive ions have to leave the cell before they open. The effect of cyclic AMP is to change their shape so that they open with fewer positive ions. So instead of having to get to this point, they'll open here, and thus we'll see we haven't gone as far negative, so we don't have to go as far positive, and our action potential occurs earlier. So with the effect of norepinephrine, we see this, instead of a longer event, so in the case of 1, 2, 3, we would get 4 or 5 beats in that time. Does that make sense? We don't have to go as far negative.
So is the negative 50, negative 55 with the norepinephrine or
Without.
Without.
So with the norepinephrine, it's about a -40. So it doesn't have to lose as many positive ions before it starts to depolarize again. So what norepinephrine does is it causes that action potential to occur in a shorter period of time. Therefore, we have more of them per minute. Okay. It also causes long-lasting calcium channels to stay open longer, and that becomes important when we talk about contractile muscle, the force of contraction. So how do we slow the heart rate down? We use acetylcholine. All right? So acetylcholine is parasympathetic, and what it does is it inhibits. It binds to an inhibitory G protein. G proteins don't always have to be stimulatory. They can be inhibitory, and it inhibits adenylate cyclase. Hence, it decreases the number of open calcium channels, and it takes longer. Right? It also binds to potassium channels, and so it makes the inside of the cell become more negative because more potassium is leaving. So it kind of does the opposite. Rather than allowing depolarization to occur earlier, it's going to cause this to happen. Now it takes longer to reach the threshold. Therefore, fewer action potentials are produced. So it inhibits formation of cyclic AMP, so the funny current channels don't open for a while, and it maintains the potassium channels open longer, so we hyperpolarize before we actually start to do our depolarization. A pattern of the sinoatrial node. That would be our standard effect. The role of norepinephrine in increasing the heart rate is going to cause this to occur earlier and faster. Going straight into an action potential here. So with norepinephrine, we see a faster action potential, which means more action potentials reach the contractile cells, faster heart rate, and with acetylcholine, the cell becomes more hyperpolarized, and therefore it takes longer. So we have fewer. It looks confusing there, but if you count the number of action potentials, there would be fewer per minute with acetylcholine. Now if we go back to the heart transplant, and this woman is climbing, the first mountain that she climbed after she recovered from her heart transplant was Mount Kilimanjaro in southern Africa. So she has no nerve innervation. As we start to do warm-ups, and we start to exercise, all right, our heartbeat starts to beat faster before we even need additional oxygen. And then as we produce lactic acid and the pH of our blood changes, and that triggers the release of activation of the sympathetic nervous system, and our heart starts to beat faster. As we actually exercise, that rate goes up. Okay? If you are physically fit, cardiovascular fitness, and you stop exercising, your heart rate should return to normal range within 5 minutes due to acetylcholine. Okay? So without either of those, her heart rate does get faster. Even though she has no direct innervation of the sinoatrial or atrioventricular nodes. Where is that norepinephrine coming from? Why would it take 30 minutes to an hour to get her heart rate up to the speed that she needs to actually do the work at the higher elevation?
Maybe the demand for oxygen? I mean, would it just be slower because it's not a...
Her sympathetic nervous system is intact.
Right, but it's not communicating with
But it's not communicating with heart. What organ is it communicating with? Very good. The adrenal glands also release norepinephrine and epinephrine. But how does it get to the heart? The blood. And that's going to take longer. Okay? Now, she has reached base camp. Or she's flying for half an hour, she's going to stop and take a rest. Is her heart rate going to drop back to normal within 5 minutes? No. Why? She has no acetylcholine. So we have no endocrine organs that release acetylcholine. So she does nothing in the blood. Okay? Acetylcholine is released from nerve endings. And there are no nerve endings for acetylcholine at her heart. And there's no endocrine organs that release acetylcholine into the bloodstream. So what she has to wait is for the half time. She has to wait for the norepinephrine, epinephrine that's in the blood to be cleared from the blood via metabolism or the urinary system. That takes a lot longer, all right, than the breakdown of the norepinephrine at the nerve endings. So she has the capability of increasing her heart rate, but not decreasing her heart rate without any other mechanism but removal of norepinephrine.
So say like she was, going up there and worked herself so fast because she literally burned out her heart. That's where it's pumping so fast.
Probably.
Because it couldn't calm down.
Yeah. If she had those stores of norepinephrine in her bloodstream. But I'm sure she would, if she, maybe we could draw blood out of her. We're going to bleed you. All right, so we haven't gotten to the contractile cells yet. We only have 5 minutes left of class, but let's just take a brief look. So here's the sinoatrial node, and here are the contractile cells carrying the electrical current through the atria. As they pass through the atria, from contractile cell to contractile cell, they will eventually reach the AV node, and the same thing will happen in the AV node that we just saw with the SA node. But right now, let's just look at the contractile cells of the atria. They look just like the contractile cells of the, come back to that here, of the ventricular system. So we'll begin with I'll review the autorhythmic cells on Tuesday, and then we'll move into this. But what's the key thing that you notice here, different from our skeletal muscle contractile cells? A much longer action potential. Okay? This is referred to as a plateau. Anybody have an idea why we would need that? While we are in our action potential, is the muscle tissue capable of receiving another stimulus? No. What do we call that in skeletal muscle? The refractory period. The absolute refractory period meant that no matter how strong it was, the muscle cell would not respond. Why is that important? This is almost as long as the entire contraction of our cardiac muscle cell. So if we look at this image here, here is the action potential of skeletal muscle and its contraction. Here is the action potential and contraction of cardiac muscle. We would be in dire straits if our cardiac muscle went into tetany. So by having a prolonged action potential, almost as long as the contraction, whereas here in the skeletal muscle it's not, we can start another contraction before the original one relaxes. And by multiple stimuli, you can cause skeletal muscle to go into tetany, all right, incomplete irregular tetany. By having the action potential last longer, that helps to prevent tetany of cardiac muscle. So on Tuesday, we'll talk about how we create that plateau.
Is tetany a cardiac muscle? Would that cause defib or AFib?
Nope, it would be a spasm.
Would be more of a spasm?
Is it, yeah, spasm, heart would remain contracted, blood flow into the heart wouldn't occur, blood flow out of the heart would be stymied because it's contracted and that initial stroke volume has occurred, you wouldn't be pumping any more out.
Is there a heart?
So it'd be like a paralysis.
Well, I mean, like as far as doing the EKG or ECG, what would that look like? It wouldn't be, it's just
Oh, well, you wouldn't see it. You would see the single contraction, and then it would just be a straight line after that. But it would be at a higher level than the resting component. Okay? So on Tuesday, go back and review those membrane changes. All right? Read through the autorhythmic cells again, because we'll review that on Monday. I'll put these slides up this afternoon. I won't delete, I'll just add additional one and I'll have new slides like that. So you can open those. I'll just add the whole new file up there again, rather than just the new slides. Okay? So remember on Tuesday, in lecture then we'll do the contractile cells. Then we'll put all of this together as the ECG. And what we see in the ECG, because that's what we're going to do in lab, is the ECG. And then we'll look at the whole cardiac cycle. We'll go back to the atrium, the heart being relaxed, the atrium contracting, the ventricles contracting as it relates to the ECG. So that won't be anything new. The cardiac cycle will not be anything new. We'll just put it together with the ECG and what's happening with. So we'll look at what we talked about yesterday, but a step-by-step event. All right? So the lab quiz will be what we covered yesterday. So the structures of the heart. There'll probably be one image from the sheep heart, but it'll be the same structures that we did with the heart model, including the coronary arteries and veins. And I will move the lecture quiz. I think it closes Sunday night.